In this unit, we're going to be talking about organic compounds. Organic compounds that are important to life. So organic means containing carbon. You'll recall from the last unit that carbon is a very versatile element. It can form 4 bonds, allowing for molecules with long chains of carbons hooked together. We call these macromolecules. And the 4 types of macromolecules are proteins, lipids, carbohydrates, and nucleic acids. These are the 4 types of macromolecules that any living thing produces in order to do what it needs to do chemically. So once again, these are the elements very important to the molecules that make us up, or any living thing: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Here you see what's called a space filling diagram. It's a 3-dimensional diagram showing the bonds, the sticks, and the different elements by different colors as balls. So oxygen is the blue one, hydrogen is the grayish one, and the black one's carbon. So here we have C, 1, 2, 3, 4, 5, 6. C6. We got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 8, 16, and 2 oxygens, which is glucose. A chain of carbon atoms with other elements hooked on. Remember, carbon has 4 valence electrons. Again, valence means outer cloud, electron cloud, electrons. And so there's spaces for 4 more electrons to be shared. Here's a simple carbon molecule, organic molecule. Organic meaning anything with carbon in it. So here's a very simple organic molecule with 1 carbon atom hooked to 4 hydrogen atoms, 4 bonds, 4 single covalent bonds. This particular molecule, carbon with 4 hydrogens, is methane. It's the gas that the bacteria in our gut produces and causes to produce that gas that when you pass gas. But as I showed you before, carbon can also form double bonds or triple bonds. Notice that in all these situations, the carbon has 8 valence electrons, and the other one either has 8 or 2 of its hydrogen. Now, carbon monoxide seems to break the carbon rule that carbon has 4 sticks. But that's just a general rule. The real rule is that carbon needs 4 more electrons in order to be satisfied chemically, which is fulfilled with carbon monoxide. It just shows that it made 3 sticks, that's all, or bonds. And carbon does not form quadruple bonds. There's no such thing as a quadruple covalent bond. Triple bonds are the biggest covalent bonds you can get. A bond, a chemical bond, stores chemical energy. You break that bond and that energy is released. Okay, a double bond is stronger than a single bond, and a triple bond is stronger than a double bond. Stronger means storing more energy. So methane is an example of what we call hydrocarbon. Hydrocarbons, by definition, only have hydrogen and carbon in them. So they're either methane with one carbon or two or three or four, etc. They can be in ring form too. These guys are in ring form. The carbon-carbon bond and the carbon-hydrogen bond. These are examples of nonpolar bonds. If a molecule only has nonpolar bonds, it is going to be a nonpolar molecule. A nonpolar molecule does not dissolve in polar substances like we've learned before. It dissolves in other nonpolar liquids or gases. Fossil fuels are examples of hydrocarbons. Fossil fuels like butane gas, petroleum. So oil, which is a hydrocarbon, will not mix with water because water is polar. Oil is nonpolar. So we talk about nonpolar things as hydrophobic. That means water-hating, water-fearing. It doesn't mix with water. If you mix oil and vinegar in salad dressing, the oil is nonpolar, and the vinegar is a water solution that's polar. So they don't mix because hydrocarbons are hydrophobic. Polar molecules, like sugar, are hydrophilic. That means they're attracted to water. They dissolve in water. Hydrophilic. But nonpolar molecules are hydrophobic. They do not dissolve in water. If a hydrocarbon only has single bonds between the carbons, then it is said to be a saturated hydrocarbon. There are no double or triple bonds between the carbon atoms in a saturated hydrocarbon. An unsaturated hydrocarbon will have at least one double or triple bond between the carbons. These double bonds cause the hydrocarbon to have a little kink in the chain, a little bend. Okay, so let's whip through these. Organic means carbon containing. What property of carbon gives the ability to form chains? Like we said, carbon has 4 outer shell electrons. Outer shell is valence and therefore has 4 openings for connecting to other carbons or other elements. Yeah, carbon forms only most of the time, well, it says most of the time, but only it forms covalent bonds by sharing electrons. So how many electrons does carbon share to complete its valence shell? Well, it needs 4 more. So we'll share 4 more electrons with other carbons or other elements. Organic means Containing carbon. Only one I see containing carbon is this one. Okay, hydrocarbons contain hydrogen and carbon only. So they're nonpolar. So letter D is the right choice. Okay, if a hydrocarbon contains only single bonds between the carbon atoms, it is called a saturated hydrocarbon. And if it contains double or triple bonds in a hydrocarbon, the hydrocarbon is called unsaturated, because you can add things to the double bond. Gasoline and water do not mix. They don't dissolve in each other, because gasoline is a hydrocarbon, it's nonpolar, and water is polar. So that's letter B. Now, hydrocarbons form the basis or the framework for the four macromolecules that are so important to living things. Okay, these macromolecules aren't hydrocarbons because they have other elements besides carbon and hydrogen, but they're mostly composed of carbon, hydrogen, and hydrogen. So hydrocarbons with other elements added form these macromolecules. If the macromolecule has a repeating subunit, it is called a polymer. And the repeating subunit is called a monomer. So proteins are polymers with the repeating subunit being the monomer being amino acids. A carbohydrate is a polymer, can be a polymer with the monomer, the repeating subunit, being a simple sugar, like a monosaccharide, like glucose. Carbohydrate could be polymer, could be like starch. Starch is made up of a whole bunch of glucose molecules bonded together. Nucleic acids, the repeating subunit or polymer is nucleotide. We'll get into all this in more detail. So here is just a simple drawing of a generalized drawing of a monomer. And when those monomers hook up, combine together to form a macromolecule, we have a polymer. So the monomer is a repeating subunit. Okay, here's a specific example. You might have seen something like this. This is DNA molecule, part of the DNA molecule. This whole thing right here. And just that little piece right there is the monomer, the nucleotide monomer. So there's a nucleic acid DNA polymer, this whole thing on the left, or part of a DNA molecule. And right here, this little box right here is a nucleotide, which is the monomer of the nucleic acid of the DNA. And you can see it's made of about a dozen different atoms, but they're just showing a simple picture right here of a nucleotide. This picture is showing some carbohydrate polymers, starch, cellulose, glycogen. Okay, starch is in flour, pasta, anything made of flour. It's a food storage molecule that plants put their glucose into, hook their glucoses up into. So right here is a glucose molecule. Each of those corners is a carbon, except for the O. So when they draw it like that, that's a ring of carbons with, you can see an O right there though. Okay, so that thing I circled is a monomer. These are 4 examples of carbohydrate polymers, chains of monomers. Starch. Cellulose is what we call fiber in our diet. Cellulose is made of glucose molecules too. Those are the monomers again. And you can see that, look at the red boxes. The little red boxes, they alternate. We can't digest cellulose. We can't. It's just like a little scrubby brush fiber that keeps our intestines from getting too many bacteria on the wall of the intestines, clogging things up, causing infections. So this cellulose, the fiber in our food, keeps those drains unclogged. And then glycogen is another polymer, carbohydrate polymer, that's how we store our glucose between meals in our muscles and liver. So glycogen, again, are glucose molecules that are hooked up in a different way than starch or cellulose. In between meals, we'll break down the glycogen to give glucose in our bloodstream to feed the cells continuously between meals. So we eat a meal, we break down starch into glucose, and then we rehook those glucoses up into glycogen, and we store that in our muscles and liver, and we break it down between meals to provide the glucose that every cell in our body needs all the time. Polymers are giant molecules, again, formed from repeating subunits called monomers. So the reaction that builds a polymer from monomers is called dehydration synthesis. Dehydration means for every monomer that's linked together, one water molecule is given off, is removed from the polymers and released to the environment. Synthesis means forming together, snapping together. So dehydration synthesis means water is released, and as the monomers snap together, as a chemical bond forms between the monomers. Let's symbolize a monomer as a happy face with an OH group on one end and a hydrogen on the other end. Notice that OH and H makes HOH or H2O. Okay, so when a monomer snaps together with another monomer, what happens is the H on the end that's going to be connected to another monomer connects with the OH, combines with the OH on the second monomer, and the OH is removed. That's called dehydration. And synthesis, because what happens is now a new bond forms between where those two were, where the H and the OH was. So that new bond is formed, making the polymer grow in size. We use dehydration synthesis when we build our molecules that we need from the molecules that we've broken down by digestion. So our cells build up polymers, the polymers that we need, our cells need, from broken polymers that we've eaten. So the monomers that are obtained by breaking down our food polymers are built together into the polymers we need by dehydration synthesis reactions. Now, when we digest, when we break down polymers, we use a process called hydrolysis. Okay, hydro means water, and lysis means breaking, splitting. So hydrolysis is splitting up a water molecule to cause a polymer to break apart. Okay, let's show that. Okay, so here's a molecule that we need to break down. And let's say we're going to break this bond here. So what happens is water molecule in the surroundings, a water molecule is split up into H, and the H goes on this end, and the OH goes on the other end. And what happens is that energy in the bond is released and breaks that bond. So the water is split into H and OH, right? Into H and OH. That's called hydrolysis, water splitting. And then that breaks the bond that existed between those two monomers. So when we digest our food, we do hydrolysis. So when we break down molecules we don't need anymore in our cells, that's called hydrolysis. So you can see that these reactions are reverses of each other. If you have a dehydration synthesis reaction and you switch it around, a polymer plus water forming monomers, then you have a hydrolysis reaction. So the dehydration synthesis and hydrolysis reactions are reverses of each other. So here's one of those comparison questions. So what is to carbohydrates as what is to proteins? Well, what are the building blocks or the monomers of carbohydrates? So here's a polymer. And so the monomer of a carbohydrate are simple sugars, like monosaccharides. One glucose molecule, for example, the carbohydrate could be like starch. Okay, so proteins, if that's the polymer, what is the monomer of a protein? Amino acids. So this would be my answer. You just tell me what are the amino acids of these two polymers. That's all. Okay, is this what kind of reaction is this? Okay, we got water. So water is a reactant. And it looks like we split the water up. If the water was on this side, which it isn't, then we know we produce water. We gave water off by the reaction. But what happened is using the water. So if you're using water up, it's breaking down the water. This is called a hydrolysis reaction. Because water is breaking down into H plus OH. Now we can prove that by balancing the equations on the reactant side, which is to the left of the arrow, and the product side, the stuff to the right of the yield arrow, the reaction arrow. So reactants, carbon, how many do I got? 5. How many do I have over here? 1, 2, 3, plus 2 is 5. Okay, hydrogen on the left side. 12 plus 2 is 14. How many on the right side? 7 plus 6. 7 plus 6 is, plus 1 right here is 14. Okay, how many do we got of what's left? Oxygen. We got one oxygen over here. We got one oxygen over here. So 1, 1. So it's a conservation of matter. No, you don't. In a chemical reaction, you don't lose or gain types of elements. But what's happened is the water has broken up, and part of it has gone to this is broken up, this polymer, and OH is connected there, and the H has connected to the other side of the broken polymer. Let's do this the easy way. I see water being produced. So water's not being broken down, it's being produced. So this is a dehydration synthesis reaction. Okay, so this guy, this OH, one of these H's went together and formed a water molecule, taking out an H and an OH. And you can see these two guys, minus the H and the OH, combined together to form this. The simple way is if you see H2O on the right side, it was produced and not broken down. Since water is not broken down, it's not hydrolysis. So the reaction is a synthesis, dehydration synthesis reaction. Water was taken out of the reactants. Or you can see that C8, the carbon backbone, was built up. It was 5. Now it's 8. So that's a synthesis, a dehydration synthesis reaction. So dehydration synthesis. So dehydration means water is produced, is taken out of the reactant. So look on the right side for one that has water in it. Okay, I only see A is having water on the product side. Okay, these two guys have water on the reactant side. So those are hydrolysis reactions. This guy right here is some other kind of reaction, but neither hydrolysis or dehydration synthesis. So my answer is letter A, again, because water is taken out of the reactants, is produced. Okay, what is true about dehydration synthesis? Water is formed. Let's see, water is formed when the monomers join. Yes, synthesis means the monomers are combining together chemically, and dehydration means water is produced. It's taken out of both monomers, parts of the water. So it has to be B. Covalent bonds are formed between monomers. Yeah, that's true too. And if you have, it's probably all the above are true. Let's just check letter A. One monomer loses a hydrogen atom, and another loses a hydroxyl group. Okay, hydroxyl group. We talked about that in acids and bases, but that's caught up. That's the OH. So that's true. One of the building blocks loses a hydrogen, and the other loses a OH group, and together they'll produce water as the two monomers snap together. So all those are true. Okay, the opposite of a dehydration synthesis reaction is hydrolysis reaction. Okay, dehydration synthesis produces water and a bigger molecule of polymer, a longer polymer. So the best answer looks like this one right here, because it has water in it. Okay, hydrolysis is when water molecule is broken down to H and OH, and these connect to the two monomers where the polymer was broken. So let's go through each one of these. The products of hydrolysis are larger than the original polymer. No, the polymer was broken up into monomers, so it can't be that one. Products of are water. No, hydrolysis is not a product, it's a reactant. So it's not that one. Two monomers. Well, not really. Two parts of a polymer. Now, if you only had a polymer with two monomers, then yeah, this would be true. Okay, so hydroxyl group and a hydrogen atom. Well, that's true. That's better. This would be, letter C would be the correct answer if you only had a two monomer molecule, and then when water was used up to form OH and H. Okay, so we have monomer 1, monomer 2, but it could be a much longer molecule that gets broken down in a hydrolysis reaction. So letter C is not always correct because you could have a much longer molecule. When hydrolysis occurs, you'll get not just two monomers, you get two smaller polymers. We're now going to talk about the 4 types of polymers that all living things need. Let's begin with carbohydrates. Carbohydrates consist of carbon and water. And that's where the name comes. Carbon, hydra means water. There's polymers of carbohydrates called polysaccharides, which we'll talk about in a few minutes. But simple sugars, simple carbohydrates or simple sugars, there's 2 types. Monosaccharides, mono means 1, and disaccharides means 2. Now, monosaccharides means 1 ring of a simple sugar. So you got the carbon backbone, but it's instead of carbon, carbon, carbon. Instead of that, it's in a ring. There's several kinds of monosaccharides. We have glucose right here. Now, each of these corners represents a carbon atom. They just don't draw it because the picture will get really messy if they did. So there's glucose and there's fructose. They're all 1, 2, 3, 4, 5, 6 carbon rings. And then there's galactose. But all 3 of these are types of monosaccharides. They're all C6H12O6, but different, what they call isomers of C6H12O6. Okay, now, those are monomers. Now, you put 2 monosaccharides together and you get what are called disaccharides. As I said, 2 rings. So if you have glucose and fructose together, you get the disaccharide sucrose, which is that white cane sugar that we have at home. Glucose and galactose bonded together makes a disaccharide lactose, which is milk sugar. It makes milk sweet. Some of you guys are lactose intolerant, and you get tummy aches because you can't digest the lactose sugar in milk products. Okay, now, those guys, these are the disaccharides right here. The 2-ring sugars. Now, when you have a whole bunch of rings connected together, we have polysaccharides. That is a polymer of that's a carbohydrate polymer. So we got, of course, starch. That's in flour. That's how plants store their glucose for when they need food. We got glycogen. That's animal starch. That's how we store glucose between meals. Glycogen is stored in our muscles and in our liver. Then we have a polysaccharide cellulose. Cellulose is plant fiber. We'll learn about that later when we talk about plants. We cannot digest cellulose, but the fiber is good for our body because it's like a scrubby brush for our intestines, keeping bacteria from growing too thick on our inside intestine walls. Okay, for monosaccharides, we're always going to have the ratio of carbon to water in the formula of the monosaccharides. Always going to be 1 carbon to 1 water. So C6H12O6, CH2O, it's 6 to 6, which is a 1-to-1 ratio of carbon to water. 6 over 6 is the same as 1 over 1, right? 2 times 6 is 12. And then 1 times 6 is 6. And then, of course, we got 6 there. So this is a 6 to 6 ratio, which is the same as a 1-to-1 ratio. Any monosaccharide is going to happen. So in this case, 3, then, and if you work it out, C3H2 times 3 is 6. O1 times 3 is 3. So you get always a 1-to-1 ratio of carbon to water in monosaccharides. Okay, I already went through this. You can read this again. This is really the only polymer, carbohydrate polymer. These other guys, monosaccharides and disaccharides, are simple sugars. I would not consider a disaccharide as a polymer. I wouldn't, because it only has 2 monomers, 2 monosaccharides. Polysaccharides have many monomers. We get our monosaccharides from fruits and vegetables that are sweet, like green peppers. I skipped this in the last frame. Notice that monosaccharides end in the suffix OSE. So if you see OSE at the end of a name, it's a simple sugar or a monosaccharide. Or it could be a disaccharide like sucrose or lactose. So what's the importance of simple sugars? We break simple sugars down into carbon dioxide and water by a process called respiration. And it extracts the energy from the chemical bonds between the carbons. We'll talk about respiration later. Also, another important feature of simple sugars is they provide the carbon we need to build other biomolecules, organic biomolecules that we need, which we'll talk about coming up. Also, simple sugars are stored in polysaccharides. So the disaccharides and monosaccharides are the monomers which build that compose the storage sugar, the sugar that is stored in, so that we always have a supply of glucose on hand. We'll just snap off the glucoses off these storage molecules like glycogen or a plant like starch. Okay, this picture shows what fructose here and glucose here. These are structural formulas. These are two isomers of C6H12O6. Same chemical formula, but different structural formulas. Now, I highlighted the hydroxyl groups. Hydroxyl groups will hydrogen bond with a water molecule. And this is why monosaccharides are very soluble in water, because water will hydrogen bond with the hydroxyl groups, the OH groups, and tear the individual monosaccharides out of the sugar crystal. So let's do some questions to review. Carbohydrate described by the formula C5H. Okay, so we always know for carbohydrates, well, monosaccharides at least, that there's always going to be a 1-to-1 ratio. So the X is equal to 5, the carbon subscript, which is 5. So X equals 5. So this be C5H 2 times 5. X has to be 5 over there too. 2 times 5 is 10. Oxygen 1 times 5. So the carbon in oxygen always has the same subscript in a monosaccharide, and the hydrogen is always twice that number. So what would be hydrogen's subscript? That'd be 10. Okay, in this one, we can take a shortcut. Hydrogen's always twice what the X's are here. So that would be half of 14. X would be half of 14. So it'd be 7. Or you can do it this way, CH2O. We're solving for X. So this would be CXH2XOX. Well, they tell you that 2X is 14. So X is equal to 7. So we can go back and say, so the formula will be C7H2 times 7, and then O7. So that's another way of doing it. In this one, I'll just do a shortcut. 6 is 2x. X is equal to 3. So this would be C3 H6O3. They forgot to tell me what to find. Okay, find the subscript X. Monosaccharide Okay, we know 3 monosaccharides: glucose, galactose, fructose. Glucose, lactose, and fructose. I don't see galactose, so it would be V and C. Disaccharides are formed by combining how many monosaccharides? Well, that's pretty easy. Di means 2. What is another name for simple carbohydrates? Carbohydrates. Well, I could call them monosaccharides or disaccharides. And sucrose would be an example of a sugar. So disaccharide. So all 3 of these, I wouldn't call starch or glycogen or cellulose, which are polysaccharides. I would not call those sugars. Those are storage molecules of sugars. So all 3 of these would be simple carbohydrates. So letter D is the correct answer. Which of the following are connected by double bonds to the glucose molecule? Well, here's a double bond. So I see carbon and oxygen connect by a double bond. Duh. Okay, we already talked about this. 3 types of polysaccharides. There's storage molecules of sugars. The important point in this frame is showing parts of a polysaccharide, in this case starch. Starch is how plants store their glucose. Okay, glycogen is like starch, but it's the way we store glucose. Animals, that's the way animals store glucose. It's more highly branched than the starch molecule, so it stores more energy per glycogen molecule than does a starch molecule. And cellulose is a polysaccharide that plants make to give them strong cell walls. Every plant cell has a cell wall around it that keeps the plant stiff so that it can grow tall. There's no skeleton in a plant, so that's why they need their cell walls. And cellulose isn't for food for the plant, it's for structure, it's for support. But some animals, well, some organisms, protozoans, can eat the cellulose and get energy from it through respiration, but we can't. But again, cellulose is good for us because it acts like a fiber that scrub brushes the inside of our intestines. Once again, just remind you, every time there's a corner, a kink, that represents a carbon atom. The picture would just be a mess if they put C's for all of that. Now notice that cellulose is very packed together and it's connected by hydrogen bonds. So you got layers of these polymers that make cellulose. Okay, so I said protozoans and some bacteria will break down cellulose. And so cattle can eat just grass. They can be totally vegetarians because in their guts are bacteria and protozoans that live there to break down the cellulose and give the cow the glucose from the cellulose so that he can do respiration to extract the energy from the glucose. This is just reminding you that in order to get energy, we need to have glucose molecules. And so we need to break down the glycogen, or plants need to break down the starch in order to obtain glucose that then can be sent to the respiration factory in their cells to extract the energy. Again, this would be a hydrolysis reaction. That's why hydrolysis reactions are important, because you got to break off those individual glucoses and send the glucose to the respiration factory. So we have some more questions here. The fundamental unit. So the monomer of a polysaccharide, which is a polymer, has to be a monosaccharide. So fructose, glucose, or galactose. Sucrose is really a disaccharide. So it's not really fundamental. It's not the simplest sugar unit. So the answer is A and B. Simple sugars do not include polysaccharides, but they do include monosaccharides and disaccharides, and glucose is a monosaccharide. Okay, starch and glycogen are similar molecules because they're polysaccharides. They're used to store energy. This is a tricky choice right here. If you look back at the pictures, starch could be unbranched. Okay, so I want to give you, starch can be somewhat branched, but glycogen is much more branched. Okay, so D is a tempting choice, but C is for sure. And they don't have two of the above as a choice. Okay, if an animal is has cellulose in its stomach, it's eating grass, hay. So the animal must be a herbivore. He's a vegetarian. That's another word. A strict vegetarian. Herbivore means they eat plants only. Carnivore means eats meat. Omnivore eats meat and plants. Decomposers breaks down anything that's dead. So the choice would be, okay, we said this makes cell walls. That's cellulose. And starch is how a plant stores its glucose. So starch and cellulose. The next biomolecule polymer that we're going to be talking about are nucleic acids. Okay, they're polymers. They consist not of just CH2O, but also nitrogen and phosphorus, NP. Okay, so they contain CH and O, just like carbohydrates do, but they also have to contain nitrogen and phosphorus. Okay, they're very important in storing genetic information. Now, genetic information are the codes, the recipes for making any protein that a cell needs. We haven't talked about proteins yet, but all our traits are because of the proteins that make up that trait or influence that trait. So I have curly hair because I have the proteins that make my hair curly. I make those proteins, and those recipes for making those proteins are stored in the genes, which are parts of the nucleic acid molecules we're going to talk about. Nucleic acids also help. There's different kinds of nucleic acids, the ones that store the recipes and the ones that carry through the baking of the proteins, the making of the proteins from the recipes. So there's other nucleic acids besides the ones that are just the library of the recipes for making proteins. Okay, so there's two types of nucleic acids: DNA, deoxyribonucleic acid, RNA. Okay, DNA stores. DNA stores the protein recipes or codes. Okay, they don't store protein. They store the information that tells the cell what proteins to make. Okay, that's stored in DNA. The ribonucleic acid RNA helps turn chemicals into proteins using the instructions from the DNA. There's three types of RNA. There's messenger RNA, tRNA, transfer RNA, and ribosomal RNA. We'll talk about this specifics later, but they don't store. RNA does not store the protein codes. It follows the protein codes in order to manufacture the proteins. So all nucleic acids consist of the monomer called a nucleotide. So a nucleotide is a monomer of the polymer nucleic acid. And here we have a picture of a nucleotide right here. Okay, so nucleotide can be broken up into three parts. A phosphate group, P connected for oxygen. A phosphate group is connected to a simple sugar, monosaccharide, either ribose or deoxyribose. And then that sugar is connected to a nitrogenous base. I just call it a nitrogen base. Okay, so three parts of a nucleotide. You should recognize this picture as a nucleotide. Once again, all these, if there's no letter at a corner of the ring, you can assume that those are carbon atoms. Okay? Okay, so here is a DNA molecule, part of a DNA molecule. Looks kind of like a ladder with rungs. Looks kind of like that. Okay, so look at where a nucleotide is. Try to circle a nucleotide if you can. Okay, so I'm going to start here. I see a phosphate group, I see a sugar group, I see a nucleotide right there. Okay, if I look at the other side, let's do the same thing. let's start here. I see a nitrogen base in red. I see a phosphate group, and in between those two, I see a sugar. Okay, so you can see that that monomer called nucleotide is repeated over and over and over. Now, DNA is double-stranded. It has two strands of nucleotides. So it's really like two polymers connected. Here's a lot of information in this picture. Okay, so on the left side we got RNA. And on the right side of the picture we got DNA. Now, RNA is single-stranded. So RNA is single-stranded. It doesn't have another strand. It doesn't have two strands of nucleotides. It just has one, as you can see, one. Now, nitrogen bases, they call them nucleobases, I don't know why. So nitrogen bases are listed. There's 4 kinds of nitrogen bases, actually 5 nitrogen bases. Remember, that's what makes up the rung of the strength of the ladder if you're DNA. Okay, so the rungs of the ladder, the things you step on, consist of 2 nitrogen bases. Okay, but you don't have them paired up in single-stranded RNA. Okay, remember, I'm looking at DNA on the right side. Okay, so let's go back to DNA. There's 4 kinds of bases: cytosine, guanine, adenine, and thymine. Okay, you should know the letters, at least, for the different kinds of nitrogen bases that can form the nucleotides of DNA. Now, the phosphate and the sugar are not going to change. The phosphate group and the sugar group are not going to change in the nucleotide. In RNA, you got the same bases almost. You got cytosine, you got guanine, you got adenine, but instead of T, there's no T. You got uracil. There's no T, no thymine, and DNA has no uracil. Again, these are the 5 different kinds of nitrogen bases that a nucleotide can have. Now, ribose is a sugar that is in RNA, whereas deoxyribose is in DNA. That's where you get the D and the R in RNA and DNA. There's no mystery about this. Deoxyribose is D missing an oxygen. So here's deoxyribose sugar in a DNA molecule, and here's the ribose sugar in an RNA molecule. So ribose means the sugar in RNA, and deoxyribose is the sugar in DNA. Each nucleotide has ribose in it if you're RNA, and each nucleotide has deoxyribose if you're. So let's go back to DNA molecule. Okay, it's double-stranded. You can see two rails of the ladder. I call them rails. The up and down things that the horizontal rungs are connected to. Okay, so those rails are sugar phosphate backbones on either side. Phosphate on the outside, sugar on the inside. Repeated over and over. Now, the rungs of the ladder consist of two nitrogen bases. In this case, cytosine and guanine. Okay, now, in between the nitrogen bases are hydrogen bonds, not covalent bonds. They're much weaker, but they hold the DNA molecule together. So hydrogen bonds attach the chains, the rails together, the two rails together. That's very important. For later on, you'll find out the DNA molecule has to be unzipped down the middle to copy the protein codes that are stored in the DNA molecule. So the hydrogen bonds, they're strong enough to hold the molecule together, but they also can be unzipped because they're weak compared to covalent bonds. Okay, you can read this. That's just what I talked about. Here they're showing the structural formulas of the 5 nitrogen bases. You don't have to identify these. You don't. But you have to know which ones are DNA and which ones are RNA. Which one is RNA. So, in thymine, of course, T is only DNA, and uracil is only in RNA. Okay, so here we're showing something very important. It's called the base pairing rules. The base pairing rules. Okay, these two strands of DNA are complementary. Meaning that the base pairs follow the base pairing rules. So the base is on, nitrogen bases on either rail has to match according to these base pairing rules. So A has to bind to T, and C has to combine to G to hold the ladder together. So you can see that that's always going to happen. If you just look at the picture, like A goes with T, C goes with G. Basically pretty easy. Okay, recall that the rails are made of sugar and phosphates, repeating. There's no code stored in the sugar phosphate backbone or the rails. Of the DNA molecule. The code, the recipe for the proteins that DNA is storing is based on the order of the nitrogen bases. So those are like the letters in the words that are telling the cell how to make a specific protein. So the gene, Gene, the genetic information is coded in the order of the nitrogen bases, not in the backbones or the rails. Of the DNA molecule is also called a double helix. Helix means like a spiral, like a spring. So double helix refers to the twisted lattice shape of the DNA. Okay, the order of the nucleotides in RNA determines the structure that the RNA molecule is gonna have. And the structure dictates the function. Depending on what your structure is, depends on how you function, how you help decode the genes in the DNA into protein. But it doesn't store any genetic information if it's RNA. Also, none of these structures are double helixes or a twisted ladder like DNA's function structure is. So, in RNA, the base pairing rules, C to G is the same as the one for DNA, but not A to T because there's no T in RNA. So that rule is changed. Instead of thymine, RNA has uracil. So there's the second base pairing rule for RNA. Now, when RNA folds up on itself, it will be base pairing, but I don't want to call that double-stranded. Once again, I'm reminding you that DNA is the library of the protein codes. It stores the genetic codes. It has the genes. RNA is the molecule that takes the recipe, a copy of the recipe, into the part of the cell where proteins are made, and it directs the making of the proteins. So it's very important that RNA decodes the code. The code is in the DNA. So let's start some review questions. Identify the monomers in the diagram below. Okay, we already did this. So here is one nucleotide right there, a phosphate, a sugar, and a nitrogen base. So the monomer, that is a nucleotide. What is the nucleotide composed of? You should be able to do this. Phosphate group connected to a sugar connected to a nitrogen base. So nitrogen base, sugar phosphate backbone is not part of a nucleotide. A nucleotide is part of the sugar phosphate backbone. So really there's two right answers here, A and B. So there should be a choice that says A and B. And I would pick that choice. A and C, I'm sorry, A and C. Okay, I'm sorry, I pushed the button after I put all the writing in. So let me explain this. How does the RNA differ from DNA? Well, there's a lot of differences, but RNA is single-stranded, whereas DNA is double-stranded. So the shape is totally different. So that's the only right choice there. But you should know all kinds of other ones. RNA has ribose sugar. DNA has the oxyribose sugar. RNA has uracil nitrogen base instead of thymine nitrogen base like DNA has. Also, the function, DNA stores the genetic information, stores the protein making codes, and RNA carries out the codes to make protein. Okay, so we already did this one, but it's asked in a different way. A component consists of a phosphate group, a nitrogen base, and ribose or deoxyribose, which happens to be a 5-carbon monosaccharide, but no glucose. So it's letter D. It's not a component of a nucleotide. Forgot to push a button. So what base, nitrogen base, is found only in RNA, but not DNA? Uracil. Thymine is only found in DNA, not RNA, but they didn't ask that. Okay, the sugar phosphate backbone. Okay, so we're looking at a DNA molecule. Let's just say, it just said in DNA. Okay, so the rails and the rungs. Okay, so the sugar phosphate backbone is the rails. So if I do a picture of that, here's a phosphate connected to a sugar. Oops. 5-carbon sugar connected to a nitrogen base. Okay, so I'm showing one little bit of one nucleotide of the DNA. Okay, now nitrogen bases in the rails are connected to each other by hydrogen bonds. That's not a covalent bond, like we talked about before, much weaker. This is a covalent bond between the sugar and the phosphate, as it is between the sugar and the nitrogen base. So hydrogen bonds are much weaker. So bonds are covalent. And covalent is much stronger than hydrogen bonds. Okay, for this one, DNA is a polymer of nucleic acids. RNA is also a polymer of nucleic acids, so that's wrong. DNA is always a double helix, true. And RNA forms many different kinds of shapes, but not double helix. Remember, the shape folds up, the chain folds up on itself to form different shapes for RNA. So that's letter B. Letter C, the bases do not form bonds. No, they form bonds with the other bonds as it folds up on itself. That's kind of tricky. We didn't talk about what an enzyme is, but RNA does not store genetic code. It carries out the genetic code instructions from the DNA. So B is the right choice. Okay, the third type of biomolecule, macromolecules, polymer, important to living things, are proteins. And proteins consist of, like the other carbohydrates and nucleic acids, carbon, hydrogen, oxygen, and also, like, at least the nucleic acids, nitrogen, but no phosphate, no phosphorus. And some amino acids have sulfur, so I'll include that. Important. Proteins are very important in all kinds of functions they have. Obviously, we'll go into more detail, but they are like machines. They can serve as pumps or motors that move things, or things that carry other molecules around, or enzymes that quicken chemical reactions. Some are structural proteins, giving structure to the organism or cell. Proteins are called polypeptides. Well, the peptide bond is the bond between the monomers of a protein, which we already said were the amino acids. So a protein is like different amino acids linked together. The amino acids are the monomers of proteins, of the protein polymer. The basic units, the building blocks of a protein. And this is called a protein, this chain of amino acids, or also called a polypeptide. Right here is a peptide bond, the bonds between the amino acids are the peptide bonds. They're covalent bonds. There's 20 different monomers of proteins, and it's weird that all life uses the same set of 20 amino acids to build its proteins. So this is like the unity that underlies all the differences in the life that exists on Earth. So another unity feature of life is that all living organisms use DNA as their genetic code holder. They also use the same genetic code in the same recipes for their proteins. And they read the code in the same exact way. All the different forms of life, whether you're a bacteria or a plant or a dog, your cells read the DNA in the same way, which we'll find out. Okay, like you had to know the parts of a nucleotide, you're going to have to know the parts of an amino acid. So here's the things I circled are the same in all 20 amino acids. Okay, amino acid has an amino group, which is NH2 right here. It has a carboxyl group, which is COOH. Okay, and they all are hooked to a carbon and a hydrogen. Okay, so I don't really call that a group, the carbon hydrogen in the middle. And there's a side chain or an R group that varies with the 20 different amino acids. So there's 20 different R groups that make the 20 different amino acids. So that's the monomer that makes up proteins. So here we have two amino acids binding together by what? A dehydration synthesis reaction. You should know that from the beginning of this unit. And in fact, any monomers that connect together do so by a dehydration synthesis reaction. Okay, in this case, when amino acids link together, the bond that forms is CN bond. The bond that forms between this C and this N of two amino acids is called the peptide bond. And water is released because a water molecule is released because it's a synthesis reaction, a dehydration synthesis reaction. So here's a better picture showing how a peptide bond will form. Again, right there's the peptide bond between this C and this N. Okay, so what comes off is water molecule, so this HO, and one of these H's combine to form water, and the rest hooks up by a covalent bond called a peptide bond. Now, proteins can be 50 to thousands of amino acids linked together. So here's the 20 different side groups or R groups in the 20 different amino acids. You don't have to memorize these. I'm just giving you a list here. A very important point in biochemistry is that the shape of the molecule, whether it's a RNA molecule or a protein, the shape determines the function. If the shape is wrong, the function of the protein will be messed up. It won't be able to do its job. Okay, there's 4 levels of structure that have to be just right for a protein to function correctly. Primary, I call it 1 degree, 2nd degree, secondary, tertiary, and quaternary structure. So the order of the amino acids is what the primary structure of a protein is. So here we have 3 different pieces of 3 different proteins. These cause the protein to have different shapes and therefore different functions, because there's different order of the amino acids. So it's not just the order, but the types of the amino acids that are lined up that dictates the primary structure. So it's the sequence of amino acids. An example of a mutation which causes the wrong amino acid to be inserted in the sequence of the hemoglobin protein in red blood cells. That protein is the protein that carries the oxygen. That one little difference in the sequence of the amino acids in hemoglobin will cause the red blood cell to sickle, become like a scrunched up sickle-like cell. And that causes all kinds of problems, sickle cell anemia, under low oxygen conditions where you're exercising, your red blood cells crinkle up, and then they clog your blood vessels, and you can die from that or go into shock. Secondary structure of a protein is due to hydrogen bonding between the amino acids, causing it to fold up, the protein molecule to fold up in a certain way. So you don't need to know this detail, but it's hydrogen bond formation between the amino acids that causes this. Now, there's two kinds of secondary structure styles. There's what we call alpha helix, like here. Here we have an alpha helix. It looks like a spiral, like, DNA's double helix, but not a double helix. Now, it's important to know that in secondary structure, the R groups are not reacting. It's hydrogen bonding between the carboxyl and amino groups. That is causing the structure to go in this alpha helix form. Okay, here's a picture of a pleated sheet, a beta sheet or a pleated sheet compared to alpha helix. It's the same thing. You can see hydrogen bonding between the amino group and the carboxyl group that's holding this together. So these are both examples of secondary structure in a protein. Again, it's hydrogen bonding between the carboxyl group and the amino groups that are doing that, causing that. Okay, tertiary structure of a protein is when the R groups of the amino acids react with each other. It could be polar bonds, hydrogen bonds between the R groups, or it just could be a non-polar R group getting attracted to another non-polar R group. But the important thing is the interactions of R groups in the alpha helix or the pleated sheet. Okay, right here you can see these flat yellow bands that really folded. Those are pleated sheet or beta sheets. and then these curly red ones are alpha helix. Is an alpha helix. But the whole thing, the whole colorful thing, is one polypeptide, one protein molecule. Well, then finally, quaternary structure consists of, you have two polypeptides, two protein, or more, it could be more, interacting with each other to make one giant protein. Okay, so there's one or more polypeptides binding together due to all kinds of bonds. It could be a covalent bond between two R groups. It could be any of the previous ones. It could be hydrogen bonds between the R groups. It could be nonpolar attractions between two nonpolar R groups. Anything between the two different chains that's or more. Anything that's connecting it and kinking it to give it this three-dimensional shape is quaternary structure. So here's a nice summary of the different levels of structure of a protein, what causes it. So the primary structure is due to the sequence of the amino acids in the protein chain polymer, and that's held together by covalent bonds between the amino acids, the peptide bonds. So that produces a single chain of amino acids. Secondary structure is due to hydrogen bonding between the NH2 and COOH groups. Of the amino acids in the single chain, forming an alpha helix or a beta-plated sheath. Okay, tertiary is one chain still, one chain, protein chain, but it's interactions between the R groups, in the chain, determining a shape. It kinks it up and folds it up into a three-dimensional shape. Globular, like a sphere. And then quaternary, 4 degree, is any kind of interaction between multiple peptide chains. So these structures, 1 through 3, were all part of the structure of a single peptide chain or protein chain. So quaternary structures, when you have more than one strand of protein, more than one protein polymer, and they're all tangled up in each other with interactions of any type, any of the types we just mentioned in one, in the tertiary structure. So, or secondary structure, because you can have hydrogen bonding between the chain, the amino and carboxyl groups as well on different chains. So the point is, those are attractions between different parts of the different chains, not the same chain. So this table gives 8 functions, common functions of proteins. There's all kinds of other ones as well, but these are ones, general ones. For structural, proteins can have just a structural function, like our hair is made of keratin protein, or the inner skeleton of a cell is made of protein fibers. That's called the cytoskeleton. When we get to the cell unit, we'll talk about the cytoskeleton. In our muscles are made of proteins that can contract when signaled by a nerve impulse. Okay, defense antibodies. Antibodies are proteins manufactured by white blood cells that help fight infections. Proteins can have a transport function. Hemoglobin in red blood cells carries gases like oxygen throughout the body so that all cells can have oxygen. Also takes away the carbon dioxide. Transport proteins can also be membrane channels, which are just proteins with a hole in it that lets certain chemicals in and out of the cell, like gatekeepers. Signaling proteins. The membrane can also have, here's a cell membrane, and here's a signaling. It receives, membrane receptors, it receives a hormone, let's say. A hormone is a chemical that tells the cell to produce a certain thing. Okay, so here is the receptor protein. Here's the hormone. The hormone makes the receptor protein turn on, activate other proteins inside the cytoplasm to make a certain protein needed, directed by the hormone to make. Talk about that later. Enzymes, very important. They're catalysts. They're enzymes or catalysts, proteins that speed up reactions. Reactions go way too slow without an enzyme to moderate it. And then some proteins are motor proteins. They move organelles through the cytoplasm of a cell. They move the chromosomes during mitosis and meiosis. So they actually move things. Or they also could be pumps. They could be like the membrane channels, but they use energy to pump things that wouldn't diffuse into the cell naturally. We'll talk about all this later. Just examples of some of the functions of proteins. And again, the codes for all the proteins that a cell or an organism needs are where they're in the DNA molecule. Okay, let's do some review questions. Glucose molecules are the starch. So these are the monomers of starch, which is a polymer. That's proteins to proteins. So what are the monomers of proteins? Amino acids. Which of the following is not a part of amino acids? Well, our group is, amino group is hydroxyl group. No. Carboxyl group. Okay, carboxyl group is COH. Amino group is NH2. The R group. You just put it in as a box. There's 20 different ones of those. There's no OH group. Which component varies between the 20 different types of amino acids? Well, that's the R group. The tertiary structure of a protein refers to its size. No. Presence of pleated sheaths. No, that's secondary structure. Is overall 3D structure? Yes. So is quaternary, though. The number of R groups it contains? No. So it's letter C. Okay, the what structure of a protein consists of a chain of one chain, a chain, assembled in a specific order. That's primary structure, the sequence of the amino acids in a chain. Hydrophobic and charge interactions that have occurred between R groups. Okay, between R groups is the key thing of adjacent amino acids in a protein chain. So, if it's between R groups, it's not secondary structure. And if it's one chain, it's tertiary. If there were two chains, it would be quaternary. Okay, so hormones can be proteins. And a hormone can signal a cell to turn on and make something, some other protein. So again, I kind of explained this before. Here's a receptor protein. That's another kind of protein on the membrane. It's in the cell membrane. And there might be a gland will make a hormone, which can be a protein. It could be a steroid lipid too, but we haven't talked about that yet. Once they connect, it sends a signal to the nucleus to make a certain protein. So signaling. Hemoglobin is an example of it transports the oxygen in our blood. Okay, the final biomolecule that we'll talk about, macromolecule, are lipids. Weird thing about these, unlike the other three, proteins, carbohydrates, and nucleic acids, lipids are not polymers. They're not polymers. They're big molecules, but they're not. Okay, the main purpose functions of lipids, they store a lot of energy, more than glycogen or starch. Energy storage. Think of fat. A lot of energy in fat stored there. All cells have a cell membrane that keeps the inner part of the cell separate from the environment that the cell is in. That outer skin, if you want to call it that, membrane, is made of lipids. Also, some are hormones. Some are hormones, like some hormones are made of proteins, and they're shock absorbers around our organs. So our organs don't get damaged if we fall down or get in an accident as much because the fat is like a cushioning. Okay, this is a reminder. Hydrophobic means the molecules repelled by water. They are not attracted to water. nor do they dissolve in water. Hydrophilic means opposite. They are attracted to water, and they will dissolve in water if they're small enough molecules. So lipids can be totally hydrophobic, like oil, fats and oils. They are not going to dissolve in water at all. So here we see like olive oil, and here's water. It just doesn't mix. But lipids can also be amphiphilic, means both. Part of the molecule is polar, and part of it is nonpolar. Or really, I should say, part of it is hydrophobic and part of it is hydrophilic. So we can think of a phospholipid, the kind that make up a cell membrane. Phospholipid, one word, as being amphiphilic. So you have hydrophobic tails, actually two of them, and a hydrophilic head. If you look at this picture down here, the hydrophilic head is due to a phosphate group, and the hydrophobic tails are due to hydrocarbon tails, carbon, 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 and hydrogen. So here's another picture. Here's a phosphate group that allows water to be attracted to it because you have O connected to, you got these oxygens exposed to attract the hydrogens of water with hydrogen bonding. Then we have they call them fatty acid tails. I just call them hydrocarbons because you can see only carbon and hydrogen are in the tails. But then we have this acid, this carboxylic acid, COOH group, carboxylic group. But that when you attach that to a hydrocarbon, then the hydrocarbon becomes a fatty acid. So those tails are hydrophobic because there's no way that water will be attracted to them because there's no oxygen to form hydrogen bonds with. So a cell membrane, we're going to learn later more detail about this, but cell membrane is a double layer, a bilayer of phospholipids with the polar heads on the outside, the fatty acid tails, or I can say the hydrocarbon tails, pointing towards the inside of the membrane. The inner layer of the membrane, the most innermost layer closest to the inside of the cell, is a polar heads again of the second layer. Okay, so we got an inside and outside of the membrane that is attracted to the water. So this is outside the cell, the water outside the cell. Then we got the water inside the cell of the membrane. Inside we got a waterproof layer, well actually two layers of fatty acid tails that keeps soluble stuff from entering the cell. Okay, again, a fatty acid. Here's the hydrocarbon tail. Then a carboxyl group at the end. makes it not just a hydrocarbon but a fatty acid. Now, that's going to connect with a molecule called glycerol, which is alcohol. You got 3 hydroxyl groups, and when that connects, we're going to get, if you have 3 fatty acids connecting to each of the OH groups on the glycerol, remember this is glycerol, H has come off because water is removed, because this is when 3 fatty acids connect with 1 glycerol. This is a dehydration synthesis reaction. So the green thing is glycerol. The 3 black ones are fatty acids. If you have 3 fatty acids connected to 1 glycerol rather than just 2, like in a phospholipid, what you have is a triglyceride because it has 3 fatty acids connected to the glycerol. A phospholipid only has 2. Okay, saturated lipids. There are no double or triple bonds in the hydrocarbon tail or the fatty acid tail. Okay, these saturated lipids are not as healthy as unsaturated ones to eat because they're harder to break down for the body and they're stored as fat. They're also solid at room temperature. Think of butter. Well, if you have a warm room, the butter gets kind of soft, but it's basically solid. Unsaturated lipids have double or triple bonds in the fatty acid tails. They are therefore liquids at room temperature, and they're healthier to eat because a double bond or a triple bond is easier to react and break up and release energy than is a single bond. Remember what we said, when you have double or triple bonds in a hydrocarbon, it's going to kink. It's going to bend that carbon backbone. Soap is an example of an amphiphilic lipid. So the hydrophobic part connects to the grease on your hands and the dirt in the grease, and then you soap up your hands, and the hydrophobic part attracts the grease, takes the grease off your hand, and then the hydrophilic part connects to the water when you rinse your hands, the soap with the grease comes off. So we see the dirt in the grease being surrounded by the hydrophobic ends of the soap molecules, and then once it's connected by all these, once it's surrounded by all these soap molecules, the other end, the hydrophilic end, attaches to the water, and it just rinses the dirt off your hands down the drain. Okay, another type of lipid that's besides oil and fats that is hydrophobic is wax. Wax remember, if I don't draw it, each of these corners is a carbon. And of course, there's hydrogens on each carbon. They're not drawing it to make it look confusing. But wax is just a big giant hydrocarbon with a O group there. It's really just two fatty acids connected to each other. And totally hydrophobic. So it protects a leaf from water damage. Too much water on a leaf, and it will be easier to be infected by bacteria or fungus. Our skin produces wax as well, to waterproof it for the same reason. Okay, steroids are hormones, but they're lipid hormones. You remember that some proteins are hormones as well. Well, steroids tend to have this kind of cool looking structure with 3 hexagons connected to a pentagon. Okay, somewhat. That's kind of the general floor plan of a hormone that is a lipid. Okay, let's finish this up with some review questions. How are lipids different from other large biological molecules? Well, there's a lot of differences, but they are not polymers. Okay, lipids can be hydrophobic, yes, like oil, fat. It can be amphiphilic, like phospholipids in cell membranes, or soap. So I guess letter C is the right answer. A phospholipid is an example of a amphiphilic. Has both hydrophobic and hydrophilic ends. Fatty acids with double bonds between some of their carbons are said to be unsaturated. Which the following is not a lipid? Well, wax is. Cellulose is a polysaccharide. Cholesterol is an example of a steroid. Triglyceride is an example of a fat. So cellulose is the right answer. For which the following is not one of the 4 major groups of molecules? Okay, what are the 4 major groups? Carbs, proteins, lipids, nucleic acids. Okay, so carbohydrates, proteins, lipids isn't even there. Nucleic acids, so glucose is not one of the 4 major groups, but it is a member of the carbohydrate family.